In this video, I'm going to show you how you can process your single-cell RNA-seq data. Here, we're going to show the first essential step when you work with 10x single-cell data, which is running Cell Ranger block in Platforma. You can see that I already have a data set added to my project, and here we have 4 samples, multiple lanes, so each sample sequenced on 2 lanes. For each lane we have different FASTQ files. I also have a metadata added to the project, which we're not going to use in this tutorial, but it will come in handy later. So essentially, after you added your samples, your FASTQ files to the project, the next thing you want to do is to run Cell Ranger if your data was obtained with 10x protocol, of course. So I'm gonna go to add block and I'm gonna find Cell Ranger block here and I will add it to the project. Now the rest is very simple. You just have to select a data set. Here I only have one, which is 10x gene expression. This is the name that I gave my dataset in the samples and data block. And finally, I have to select the species. In this case, this is a mice RNA-seq. So I'm going to select musculus and click run. Now, in the background, Cell Ranger will run and process all of the samples. While it's running, you see that right now it's all complete immediately, but depending on the size of your data set, it might take some time. While it's running, you can always click here on the open for each sample, and you can see the logs that are updated in real time. And after the analysis for each sample is complete, for this sample you will see the visual report with the alignment rate, which shows how many reads were aligned to exons, introns, or how many reads were not mapped to anything, which we see here in the red. And finally, we also have a Cell Ranger web summary. And this is the standard output for Cell Ranger. So we see the estimated number of cells, mean number of reads per sample, and all of the different statistics that Cell Ranger provides by default. You can also go immediately to gene expression, and here you can see standard t-SNE plot. You can see some log 2-fold change p-values for different clusters. So you see already clusters and a table with all of the genes per cluster and their p-value for the difference between clusters, as well as sequencing saturation and median genes per cell, which is again just a standard statistics for Cell Ranger. Now, that is pretty much it. You can check this for each sample, and after this block finishes running, you can proceed to next steps in downstream analysis of your data.